What are targeted oncolytic peptides and why do they represent a breakthrough opportunity in oncology?
Well, I'm really glad you asked. So, targeted oncolytic peptides are basically,
Your mission emphasizes preserving patient quality of life. How does this principle influence your scientific and clinical decisions?
it is the center of all of my decisions. At the end of the day, like I said earlier,
Welcome to Life Science Insights podcast. I'm your host Sandal Karnanidhi, and in today's episode, I'm excited to be joined by Dr. Stanley Lewis, the founder and CEO of A28 Therapeutics, physician and scientist whose career spans translational medicine, targeted cancer therapies, and biotech innovation. Dr. Lewis has spent decades advancing drug development from bench to clinic, combining deep clinical insight with the strategic leadership to tackle some of the most complex challenges in oncology. In today's conversation, we'll explore his journey from medicine to entrepreneurships, how translation science is reshaping cancer care, and what it takes to build biotech companies that are both scientifically rigorous and patient centered. Welcome to the show, Dr. Lewis. It's a
thank
pleasure to have
you.
you here.
I'm glad to join you, Satya.
Can you start by telling us a bit about your background?
Sure. I'm an internal medicine physician by training, and I trained at University of Texas Medical School at Houston, where I did my internship, residency, fellowship, and then joined the faculty there for several years before I entered into the biotech world, where I went to a small company where we were developing a treatment for HIV disease. That company was acquired by another company, and I continued to develop the drug that I left academia to work on, and we finally got it approved as Trogarzo, the first monoclonal antibody for HIV. And then I came out here to California, where I am now, to work on a drug for treatment of influenza. But then COVID came along and much cured the flu, so we couldn't finish developing that product. And that's where I was fortunate to stumble upon the product that I am developing today, which is a targeted oncolytic peptide. And I know that word's probably not that familiar to your audience, but essentially we have a peptide with a targeting unit and a peptide payload that is used to kill cancer.
Yeah, that's an inspiring journey. You had an extraordinary journey from physician and clinical researcher to both biotech CEO. What inspired you to build A28 Therapeutics and take on this path?
You know, there are really several spots along the way where I had to make decisions that ultimately led me to where I am now. The first one I referred to already was going from academia and seeing patients and teaching students and doing some clinical research to going into the biotech space, which is a lot different. And the thing that really inspired me to do that was the patients. It was the early days, and the only medicines that we had to treat patients were very, very toxic. And I kept saying, someone needs to come along and create a new drug for these patients that's not so toxic and can be better tolerated. And then I realized that no one was going to come along and do that. I needed to do it myself. So I left academia and I went into biotech. And then once I was in biotech, I realized that to really do medicine the way I wanted to do it, to really do drug development the way I wanted to do it, and prioritize the things that I wanted to see prioritized, I was going to have to be high enough up in that company to be able to influence the way the company moved. And ultimately, I realized that the only way you can be high enough up is to be the CEO and the founder. So therefore, I started A28 Therapeutics.
Okay, that's an interesting journey. Looking back, was there a pivotal moment in your clinical or academic career that pushed you towards developing next generation cancer therapeutics?
Yes, I would say that over the time, I sort of developed a philosophy as it comes to drug development. I took my Hippocratic oath very seriously. First, do no harm. So any drug that I want to develop has to have an extraordinarily well-tolerated safety profile. Secondly, I believe that nature will give us clues as to how we should approach disease, how we should approach shifting patients from going down the wrong path to going back down a path of better health. And then finally, I mentioned I'm from Texas. I like to keep it simple. So keep it simple and straightforward, as they say. And that that philosophy on how you develop drugs has always sort of stayed with me. And I feel like cancer therapeutics are in very similar place as patients are suffering from the side effects of chemotherapies and other treatments that make them oftentimes wonder, is it better to take these drugs and lose my quality of life? Or should I just take my chances with the cancer? So that's really one of the reasons why I feel like we needed better therapies. And the current therapies that we have right now, while they are getting better, I still think there's lots of room for improvement.
Okay. For those unfamiliar, what are targeted oncolytic peptides and why do they represent a breakthrough opportunity in oncology?
Well, I'm really glad you asked. So, targeted oncolytic peptides are basically peptide um, peptide targeting units that bind to receptors that are found on the surface of cancer cells, and a peptide payload, in this case, a lytic peptide payload that can lyse and destroy the cancer cell when it gets close to it. So, targeted oncolytic peptides are a lot like ADCs or antibody drug conjugates,
Okay.
only instead of using an antibody, we use a peptide to target. Instead of using a drug, we also use a peptide as the payload.
Okay. How does A28 Therapeutics approach precision oncology differently compared to conventional cancer treatments?
Well, at A28, we don't make bespoke therapies for each patient. Instead, we make one therapy that when the patient takes it, it destroys that patient's cancer.
Okay.
The way it destroys that cancer results in the release of tumor-associated antigens from that patient's own cancer. Therefore, those antigens can train that patient's immune system to fight that patient's cancer. So it's not like cancer vaccines where you give off-the-shelf antigens and hope that that will work to train the immune system to fight that patient's cancer. No, instead, we destroy the patient's cancer directly, and then that releases those tumor-associated antigens, and that can help the patient immune system fight the cancer throughout the body.
Okay. If we look ahead 10 to 15 years, what does the ideal landscape of cancer treatment look like and what role do you want A28 Therapeutics to play in shaping it?
It is the center of all of my decisions. At the end of the day, like I said earlier, first do no harm is the guiding principle, the first principle that I take into drug development. I understand some therapies are going to have side effects. Some therapies are going to be difficult to take, but I choose to spend my time and my effort, my energy developing therapies that will be safe, well tolerated by patients, and preserve their life.
Okay. The translation medicine is often about bridging discovery and patient benefit. What are the biggest translation gaps in oncology today? And how is A28 helping to choose them?
Well, the biggest gap today is the same as it's been sort of all along, is that what you see in the test tube
Hmm.
and then what you see in animal studies doesn't always translate into something that's safe and effective in patients. And so we're at A28. I think the probably the therapeutic class of molecules that is most directly translatable from preclinical animal models into patients is
Okay.
antibiotics. And antibiotics, such as vancomycin, are lytic peptides. So we're just developing a different kind of use for those same types of medications that have been very effective at making that leap from animal to man.
Can you walk us through the scientific journey of A28's lead candidate from concept to clinical stage development?
Wow. Well, that's an interesting journey. So, A28 was created by Dr. Bill Hansel at the Pennington Biomedical Research Center in Baton Rouge, Louisiana. And he was a large animal physiologist, reproductive physiologist, and he noted that cancer cells have the luteinizing hormone releasing hormone, or LHRH hormone receptor, on their surfaces. And we don't know why those are there. He also noted from going to a lecture on insects that lytic peptides are useful as immune system molecules that help to kill foreign invaders. So he came up with this idea of, why don't we combine this LHRH ligand that will bind to the cancer's receptors with this lytic peptide that can actually destroy the cancer cell by lysing it. And that's how the molecule was ultimately created. And this was then tested. It was out licensed to a pharmaceutical company, and then it was tested in a phase 1 clinical trial where it demonstrated great safety, with the only side effect being a transient and very treatable rash. But it also was able to slow down and stabilize the disease. The molecule was then advanced into a phase 2 clinical trial in patients who had ovarian cancer. Ovarian cancer was selected because Dr. Hansel's wife had died of ovarian cancer. And so he really wanted to fight that disease. In retrospect, chances are probably would have been done better had he selected hepatocellular carcinoma, because this particular molecule, it accumulates and persists in the liver for 3 days, giving you this high exposure of the drug in patients who have liver cancer, or in patients who have cancer that metastasizes to the liver. So we were able to actually look at those patients who had ovarian cancer and liver metastases and see a very resounding efficacy signal. And again, the only noteworthy side effect was really this transient and treatable rash.
Cancer therapy development is exceptionally complex. What unmet clinical needs are you most focused on addressing first?
Well, I think the unmet need that we have is particularly in patients who have advanced disease. Patients who have developed liver metastases in particular really don't have a whole lot of options. There are no drugs that are approved for patients specifically that have liver metastases. And our drug, AT101, has this unique biodistribution such that it accumulates and persists in the liver. So it makes it a perfect drug for treating those patients who have advanced to stage 4 or at later stage.
How do you think targeted oncologic peptides will change the standard of care over the next 5 to 10 years?
Well, I think the wonderful thing about these types of drugs is that fact that they are so well tolerated. The reason why I believe that oncolytic peptides can be a part of every regimen that patients will take to try to treat their disease, as long as the patients have a receptor that we can attach our drug to, we can lyse that cancer cell of the cancer. It's not dependent on any specific pathways that the cancer cells utilize. It really is very simple. Bind, assess if it's a cancer cell, and if it is, destroy it.
Okay. What are the early signals or data have been most encouraging in current clinical programs?
The signal that I think is probably the most encouraging to me is the safety signal. That is, that it has this very well tolerated safety profile. But in terms of the efficacy signal, it is this hepatotropic nature of the molecule, in that it goes to the liver and it fights cancer in the liver. And if your disease, no matter where it starts, and it metastasizes or spreads to the liver, that means that you're stage 4. That means that your prognosis is going to be very bad. So being able to treat patients who have gone to this late stage of the disease is really where we believe our drug will shine and make a huge difference.
Okay, good. You led organizations through pivotal moments like large financing, regulatory milestones, and global launches. What leadership lesson have stayed with you?
Well, the most important thing I think when you look at a company is its people.
Okay,
And
yes.
being able to lead requires you to be able to listen.
Okay.
And listening to the folks that you're working with, the folks that are counting on you for that service, that leadership service,
Mm,
is huge.
okay.
That's the only way I find that you can develop team is to develop a keen sense of listening.
That's really a good thing to say.
Yeah, most people think of leadership as talking
Yes.
and telling people what to do. It's actually quite the balance of listening, understanding what the issues are, and then making the decisions.
Biotech leaders often balance scientific risk, operational risk, and patient urgency. How do you navigate that tension as a CEO?
Well, I'm learning. It's a that's the real task of
That's
being
true.
a biotech CEO is understanding that you have this resource limitation. You can't pursue every avenue that you perhaps would like to pursue. You really
Okay,
have to do what's best for the product.
right.
So as a physician, the patient was the person, sitting in front of me with that gown that opens in the back.
Okay,
When as a biotech CEO, my patient is this product, it is the drug. And so I have to do what's best for that drug, both scientifically, as well as meeting all the needs relative to the financing and relative to ultimately what is going to be most commercially viable and what's going to do the most to help patients. So that's always a tension.
okay.
And the best way to sort of navigate it is to first be aware of it and then pick your true north. Your
Mm,
true north should be what's best for the patient.
okay, okay. What advice would you give to emerging biotech founders working at the intersection of innovation and patient impact?
I think the best advice I can give to any founder of any company, quite frankly, any entrepreneur, is you can't do it by yourself.
That's
You
true.
have to get out there and you have to make friends and you have to talk to people. The answers that you look for to the problems that you have, to the issues, to your concerns are actually out there, but you'll never know unless you get out and engage. I oftentimes say that preparation is to performance as circulation is to luck. So the more you get out, the more you circulate, the better it'll feel like luck, but actually you're making your luck because you're talking to people, you're sharing your stories, and you're listening to theirs. And from those, you will find the answers that you seek.
How have you combined experiences across HIV, viral infections, and cancer that influenced your philosophy on innovation and therapeutic design?
Oh my gosh, I'm so glad you asked that question. You know, infectious disease and cancer are very much alike.
Mm,
They both represent a defeat of the immune system.
okay,
And so once you appreciate that, you can start to apply some of the lessons that were learned from antibiotics and some of the lessons from antivirals, which I've developed before, and you can apply those principles to cancer. One of the main ones that I think is very important, and we're starting to embrace this in oncology, whereas we've embraced it in HIV care and certainly in bacterial infectious disease for a long, long time is the idea of combination therapy. If you want to control the cancer, you must hit it with multiple drugs
mm,
at the same time, no sequential monotherapy at the same time, so that you control the heterogeneous mixture of mutant cells that make up the tumor. So it really doesn't make sense to sequentially use drugs and then wait until the cancer starts to come back and then you switch to some other drug. We learned a long time ago in infectious disease that that is not the way to approach this. you have to hit that the tumor with hopefully at least two, but maybe three drugs that are able to sort of hem it in and prevent it from continuing to expand.
Okay, nice. What technological or scientific trends excite you the most in the future of oncology?
Well, targeted oncolytic peptides excite me the most. I think that this idea, this motif of a targeting unit and a payload
Hello.
is going to be something that translates until we can find a cure. The idea that you can limit the toxicity of the medication by concentrating it at the site of the cancer is something that is welcomed and in some ways revolutionary in this space. And then what payload can you use that's going to directly destroy the cancer, but also recruit the help of the immune system? That's key. We will need cancer if we keep giving drugs that destroy the cancer, but also destroy our immune system at the same time. Your immune system is your friend. We need to enlist its help in getting rid of this disease.
How about AI? Does that excite you?
Oh, well, AI is really everywhere. And at A28, we're using AI to predict our clinical outcomes.
Okay.
So we have engaged with the GATC Health
Hmm.
that has taken all the data that we have, both internally, proprietary, confidential data, as
Okay.
well as all the data out in the universe that's published on both our molecule, our target, our disease, the indications, etc. And after analyzing all of this data, we were able to make a prediction as to whether our next clinical trial would be successful. So AI is giving us lots of confidence that we're on the right path and that will ultimately be successful.
Okay. Good.
But yeah, there's lots of different ways AI can be used
Yes.
in medicine. I think once we've proven the concept first molecule, we will definitely use AI to tweak that molecule and design new molecules on that same platform that will be able to fight not only the cancers that we know we can fight that have the LHRH receptor, but perhaps other cancers that exhibit different kinds of receptors.
Are there emerging biomarkers, delivery mechanisms, or therapeutic modalities that you think will dramatically change cancer care?
Yeah, I think what's really going to change is that we're going to go away from the therapies that we've used and traditionally used for cancer, simply because we all know they're archaic and they're too toxic. I mean, we all know that. Physicians hate prescribing them. We hate having to tell patients, oh no, you're going to have to take chemotherapy. We're going to have to move in the direction of treating patients with medications that they can tolerate. I think that's dramatically change. Now, what do these cancer drugs of the future look like? Well, I think they look a lot more like targeted oncolytic peptides. Unlike antibody drug conjugates, which sort of built off that same motif, antibodies are very expensive. And, our healthcare system is already overrun with high cost. So those molecules will limit or will be limited by their cost. And likewise, the drugs that we use in these antibody drug conjugates, well, guess what? They're the same old chemotherapy payloads that we were using before. So they don't necessarily represent that huge of a leap in terms of how the cytotoxic agent that kills the cancer. So what we're hoping for is that we'll find better, safer, more tolerated, less expensive drugs to be able to fight the cancer, both by having a peptide as a payload and a peptide as your targeting unit. I believe this is the future of cancer.
Okay, good. If you look ahead 10 to 15 years, what does the ideal landscape of cancer treatment look like? And what role do you want A28 Therapeutics to play in shaping it?
Oh, first of all, I would like A28 to play a couple of roles. I want to introduce these TOPS molecules so that we can about developing drugs. Maybe we don't have to have such, sexy science. Maybe we don't have to have such complex targeting, such complex approaches to trying to treat cancer. Maybe the simple approach is better. So that's
Okay.
one thing I want A28 to do. The second thing I'd really like A28 to do is to help drug developers eyes to different ways of approaching cancer. We are very much a me too, me three kind of industry.
Okay.
And I'd love to see more innovative ways being brought to the fore earlier instead of just continuing to make incremental changes. I want to see breakthroughs.
You often speak about simplicity and humanity in medicine. How do these values show up in your work today?
Well, I think that it's the driving force behind A28 Therapeutics. Our mission, our vision is that patients don't have to fear cancer, nor do they have to fear the treatments that we use for cancer. I think that a big part of what makes cancer so daunting is this idea that we have this terrible disease that we don't have a cure for, and you're going to be tortured before you eventually die from this disease. Well, that's just not a good. Those are not good options for patients. we need a better option for that. And so I really focus on how do we create the quality of life for the patient, and then how do we extend that life for the patient in hopes that they will allow their own body's natural immune system to ultimately overcome the disease instead of the other way around. That's how I see us being able to transform cancer care, and I go to work every day to see if I can make that happen.
For patients and families battling cancer, what message would you like them to take away from the work you're doing at A28?
I'd like them to take away these three words. We hear you.
Okay.
We hear you. And we will make medications that respond to your needs, not just to stop the cancer, to cure the cancer, but also in that process to leave you with the quality of life that you can enjoy the time that you have.
Okay. That's all we have for today. This has been an incredible, insightful conversation. And what stands out most is that how you always keep patients front and center, making sure the science leads to real-world impact. Thank you for sharing your experience, your vision, your thoughtful approach to advancing cancer therapies. We truly appreciate you joining us today.
Thank you, Cynthia. Thank you.
Okay, let's do the pause.